sometime in your life. Okay, so if you don't take these out properly, it's a problem. Okay, so here's a lot of these guys here. We're back to our old picture here. We talked about all these introns and all these exons. Here's the primary transcripts. The question is why? Why do cells go through all this problem? Why do they have to make this big RNA that you have to exert all that effort, use all that energy, use the hundreds of thousands of little molecules in order to take it out just so you can degrade it? Why does that happen? Why? Does anybody have any ideas? You should. Go ahead.
It's allowed for variability, so like one gene produces more than one.
That's one thing, but and that's the second one. I said to allow for variability. Do you know what we call that? I'll show you a slide in a minute. What's the other one? Regulation? Well, regulation's up in here, and that's why the converters are so complex. And it also, you have to process in order to get them out. So there is a level of regulation, but it's not quite as much. Evolution, right? Think of how bacteria evolve. Bacteria have this little tiny chromosome, and they have all of these point mutations that result from errors in DNA synthesis, mutations that occur with outside things, right? And if that mutation is advantageous, then it's essentially cloned within the population and that organism evolved. If it's not advantageous, that particular bacteria dies. You know, if it's lethal, it's not advantageous, that guy dies and that mutation is not incorporated into the population. Why can bacteria use point mutations like that? 2 reasons. I mean, that's a lot of experimentation, right? To have a single point mutation in your DNA to try and find something that makes your life easier. That's kind of a hard way to go about it. Well, they can do it for 2 reasons. One is they grow so rapidly. So they can try an experiment like that every 30 minutes. They can try again. If it doesn't work, they try it again, right? And what's the second reason? There's so dang many of them, right? In a milliliter, you heard, you can go, but in a milliliter, we can grow downstairs in the fermentation plant, we can grow 30 billion bacteria cells in 1 milliliter. And that's just the way we can grow them in a fermenter downstairs. So there's not only they're fast, but there's trillions of them. So if an experiment doesn't work, right, if it's lethal and the guy dies, the rest of them say, thank God there's more food for us, right? So they just live on, and they're experimenting more rapidly every 30 minutes to try and make their life better. So, so bacteria evolve very quickly that way. How about you? Right? There's not even 100 of us in this room. So our populations are nowhere near as big as bacteria, right? Population of the world is not what there is in a mill of bacteria. So populations are much smaller. And secondly, hopefully, most of you don't start experimenting until you're 20 or so, right? And if it doesn't work, it's a big effect on the population. And so it's weird for us to have point mutations. Do we? Sure we do. All of these inborn errors in metabolism, all these things that are inherited like beta thalassemia, those are mutations, those are experiments, but they happen slowly, Sickle cell anemia is old, long before any of us. Long before anybody long. So these mutations aren't very happy. But what about if you could evolve where you could take a function and take it from one protein and put it to another? What if this particular intron right there was a DNA binding sequence, a binding sequence of amino acids that bound to a DNA sequence? And now you have another protein, another transcription factor that you wanted to bind to the same one. Well, instead of evolving sort of on that particular transcription factor, you just borrow this one. You take that one out, add it to yourself, and now you can bind to that same piece of DNA. And that's what these are for. We evolve by taking each of these, taking a exon that has a particular function. These exons are domains that have certain protein functions. One binds ATP, one binds DNA, one binds NAD, one binds, and we use them over and over again. Right? If you look at a protein structure, you see in these structures they define things and they say, okay, this guy has 6 what do they call them? IgG domains. There are 6 folds that look just like an I fold that's found in an antibody. Well, they're not antibodies. They're matrix proteins, right? They're an extracellular matrix, but they use that same structure. They've borrowed an intron and then, or an exon, and then actually duplicated it. Anyway, evolution. I'm going to move and we'll finish. I guess I won't tell you about that, but I do want to tell you about that. Okay, so the other thing that we talked about, and that's what he mentioned here, is variability, right? So we only have, when I was first a student and we were asked how many mRNAs there were, people were doing experiments to find out how many mRNAs they were. You isolated methods, you did a hybridization with DNA, and you sort of counted how many hybrids you could develop, and the answer was like 150,000, 200,000. And so we thought there were 200,000 genes in the mammalian, in the human genome. How many are there? There's only 20,000, right? Were we that far off? Were we that screwed up? Were our techniques that bad? No. It turns out we were pretty close. And the reason is because there's a method that's used by cells, which he described, and that is that you can have variability of transcripts from a common gene. We call it alternative splicing. Right? So proteins can evolve so that if you have each of those numbers as an intron or an exon, I hope you're reading right. So by alternative splicing, what you can do is you can make different proteins. This is for a gene called troponin T. Troponin T is involved in muscle. Remember, it's the guy that binds calcium in skeletal muscle and allows it to interact with actin and myosin and contract. Troponin T. Well, the troponin T in a muscle cell, a striated muscle cell, is slightly different than a troponin T in a heart cell, is slightly different than the troponin T in a brain cell, is slightly different than a troponin T in a fibroblastic cell, a single cell that moves around, and they use actin and myosin, right? It's slightly different than the myosin that's used in myosin motor proteins that move stuff down microtubules like a railroad track that you learn in cell biology. This is how it happens. All of the troponins there have the first 3 exons, 1, 2, and 3. They all have exon 9, 10, 11, 12, 13, 14, 15. They all have either exon 16 or 17, and then they have 18. And then any of the 32 possible combinations with 0, 1, 2, 3, 4, or all 5 of the exons 4 through 8. So they have these here, they can have 1 of them, 2 of them, 3 of them, 4 of them, 5 of them. Right? So they alternatively splice so that they include exon 1, 2, 3, maybe 7, and then 9, 10, 11, 12, 13, 14, 16, and 18. And the other one's slightly different. So all they have to do is develop a mechanism for processing them slightly different, processing out intervening sequences. And essentially what happens is we just have proteins that cover up the recognition sites, right, of a particular intron. So it's not there. If you cover up, if you start with this 3' or 5' side of the intron here, and then you cover up these end sites here, well, then it goes to this end site here, so it cuts it all out, and you have a different message. Turns out that probably 70% of all of the genes in our genome, in the human genome, code for messenger RNA that can be alternatively spliced. Sometimes only 2 variants, sometimes 7 variants, sometimes even more variants. Averaging something like, well, figure it out, 150, 20,000, about 7. So it averages about 7 alternative splicing processes for each messenger RNA. Right? That's the difference between the 20,000 genes that you guys counted because you sequenced the genome and the 150,000 that I counted because I didn't know how to sequence. We just did a hybridization. But the numbers were pretty good. There are 150,000 messenger RNAs, but there's only 20 genes because the product of those 20 genes, the primary transcripts, can be alternatively spliced and set an average of 7 different ways each. Sense? Okay, so there's a couple more slides on here. You can ignore them. We didn't talk about them, we can ignore them. They just address the question of other proteins that help recognize intervening sequences.